Welcome, everyone. Welcome back from the break, and welcome to the Scientist and Clinician Short Talk section of the conference. We have a great line of speakers with exciting new research developments to talk about, so I don't want to spend too much time talking about logistics. We can dive right in. But for some logistics, each presenter is going to have about 10 minutes for us to play their presentation, and then about 5 minutes for some Q&A afterwards. Once we've gone through all of our speakers, they will all collectively come up at the end for any remaining time we have for more questions, probably about 10 to 15 minutes, depending on time. And as a reminder, just like our last session we saw with Dr. Chung and Dr. Boyle, as we're watching these presentations, if you have any questions, please put them in the Q&A tab on the right of your screen. When it comes to the Q&A session, I will be fielding them. And we also have some questions that were pre-submitted in advance that we can discuss as well. As a reminder to speakers, when it's your turn to come up for the Q&A, all that you need to do is just turn on your camera and microphone, and you will pop up on stage with me to discuss your Q&A. So with that out of the way, let's move on to our first speakers. We've got Dr. Michael Silverman and Troy Collins, PhD student and lab member, both at the Silverman Lab at Simon Fraser University.
Hello, I'm Michael Silverman, and I'm going to share this presentation with a PhD student in my lab, Troy Collins. I will give a very brief introduction to our lab and then turn this over to Troy. First, I want to thank the KIF1A community, and especially the patients and their families, for the opportunity to work together to advance KIF1A research. This work has given us meaning and urgency like we've never experienced, and we hope that we can contribute to the understanding of KAND and eventually a cure one day. So thank you. We're based at Simon Fraser University in suburban Vancouver, perhaps a school you've never heard of, which frankly I hadn't either until I applied for a job here. However, we're a large PhD granting institution with over 25,000 students and quite similar to many state universities with regard to academic programming. And as you can see from this picture, we have some great mountain and ocean views from campus. So we, like many others, want to understand how intracellular transport supports nerve cell development and function, and specifically how KIF1A variants might alter the distribution of essential synaptic proteins that then may contribute to KAND-related symptoms. And this idea is depicted on the left side of the screen. On the right side of the screen, from a technical standpoint, we characterize the trafficking of fluorescently tagged KIF1A cargo in living neurons, such as neuropeptide transport pictured here. From these and other data, we can start to understand how KIF1A variants affect neuronal architecture and function. So now I will turn the presentation over to Troy, who will just describe a project that is underway in our lab. And thank you again to the KIF1A community.
Thank you, Michael. Hi, my name is Troy, and I'll be talking to you today about cargo transport in R203S KAND, patient-derived induced neurons, and share with you our first steps on the transport path of studying KIF1A. So some context for my discussion, for my presentation today, be looking at whether cargo is an important part of studying KAND. Then we'll get into the specific variant of KAND that I'll be studying, R203S. And then rounding things out with my project overview, beginning with culturing stem cells to neurons, examining cargo localization to KIF1A, and then finally examining live cell imaging and cargo transport analysis in a more dynamic view of KIF1A. So, is cargo important for studying KAND? Well, we know that kinesins walk along microtubules inside axons from proximal areas close to the cell body out to distal areas in the axons, going to places such as synapses, and along with them carrying their cargo. In KAND type KIF1A, the motor protein has certain dysfunctions. And we know from previous research that a dysfunctional kinesin is found more proximal to the cell body. Functional KIF1A requires cargo binding, microtubule binding, proper ATP hydrolysis, and proper motility. And dysfunctions in any of these properties may lead to cargo mislocalization as well. So the specific cargo that we're interested in examining include dense core vesicles, which contain neuropeptides such as BDNF, brain-derived neurotrophic factor, and VMAT2, and synaptic vesicle precursors, which contain synaptophysin, synaptotagmin. Now, cargo mislocalization can lead to cellular dysfunctions, such as dysfunctional synapse formation and function, as well as disrupted neuron survival. So BDNF is a particular cargo of interest because of its role in neuron survival. Without it, neurons have a tough time surviving. We also know that these cellular dysfunctions can lead to neurodegeneration, neurodegenerative diseases such as Alzheimer's, Huntington's, and Parkinson's. The specific R203S variant, the specific variant of KAND that I'll be looking at is R203S. It is one of many motor domain variants, which we know from the literature can result in more severe pathology. They are also some of the more well-studied and characterized variants of KAND, the ones that happen in the motor domain. The cells I'll be working with are human-derived induced pluripotent stem cells, courtesy of Wendy Chung's lab over in Colombia and acquired through the Coriell Institute. The patient for which these cells come from is heterozygous and mosaic for R203S, which means we have two cell lines. We have a wild-type control and cells with the R203S variant. So we have an isogenic control, and we can compare these two cell lines. So an overview of my project. First, I'll be culturing these stem cells to neurons, and along the way characterizing the neurons and also each precursor using immunocytochemistry to validate their phenotype. Then I'll be comparing KIF1A and cargo localization using immunocytochemistry. And finally, looking at more dynamic aspects of KIF1A with live cell imaging techniques and transport analysis of a specific cargo, BDNF. This image is a timeline of the protocol we're using from Stem Cell Technologies. As you can see, it takes 25 to 28 days just to get cells beginning the differentiation of to a neuronal fate, and then they need to mature. So this is a bit of a lengthy process before experimentation can even begin. So the very first step of the sort of preliminary stage of culture is generating these iPSCs, these stem cells, growing them out and confirming their identity. So this is a SOX2 stain of stem cells that I have, one of several phenotyping stains that I'll be doing with the stem cells. And then next step will be to differentiate these into neural precursor cells, confirmed by a Nestin stain. Again, there'll be other stains that I will do to confirm their identity further. But this is yet one more step along the way before we get to the next step of differentiating them into full proper neurons. We have previous experience in the Silverman lab, growing out human neurons. An image here, for an example, are human neurons stained for dendrites and axons. And again, I want to underline the lengthy process of growing these out and getting them to differentiate and then mature. So after that 28 days, even though they're in the proper medium for neurons, we then want them to mature for about 30 days before we actually begin experimentation. So the second aim is to look at cargo localization to KIF1A. Again, our lab has extensive experience and background with localizing cargo using immunocytochemistry. In this image, this is a rat hippocampal growth cone, and in green, you can see dense core vesicles in this distal region of the neuron. And my hypothesis is that the R203S group will have cargo more proximal to the cell body than in the wild type group due to the dysfunctional KIF1A. My third aim is to look at live cell imaging and cargo transport analysis. ICC gives us a snapshot of what's going on with cargo, but live cell imaging gives us the full range of what's going on. We can look at the more dynamic aspects of transport, such as directionality, velocity, and run length. Here are neuropeptides being transported along an axon. That green arrow is a pterograde movement, and the red arrow is a neuropeptide moving in the retrograde direction. So to wrap things up, I invite you all to stay tuned. We're very excited to kick off our research. Culturing neurons, as I said, takes time, and we're really excited for the actual experimentation to begin once that's complete. And I want to round things out by saying that a comprehensive examination of cargo transport in particular will give us a more full understanding of the direct effects of KIF1A dysfunction on neuron health. And finally, I'd like to say thank you first and foremost to the KAND patients and their families. It's an honor to be a part of your journey. And thank you to KIF1A.org. The cells that we acquired were funded with a mini grant that we were awarded from them. So thank you, and for putting on this wonderful conference. And thank you to the wider KIF1A research community, as well as the members of the Silverman lab and Dr. Silverman himself. It's exciting and to be part of a larger effort for a common goal as part of my PhD thesis. So thank you.
Thank you very much, Michael and Troy. If you want to hop on stage for a question or two before our next speaker, all you need to do is turn your camera and microphone on at the bottom of your screen. Welcome.
Hi.
Thank you for your talk. Hi. And I want to say a big thank you to your group for really being one of the first groups to reach out and engage with the patient-derived lines that are now available at Coriell. It's so extremely valuable to gain more information about those lines. And the first question I want to touch on, which was a pre-submitted question, is talking about this process of differentiation. And as you mentioned, working with these lines takes a lot of time. And we ultimately want to get to that point where we're doing experiments with this finalized differentiated neurons. But I wonder, is there anything that could be learned during this extensive differentiation process? Are you seeing any differences between your wild type line and the R203S line as you're pushing them through these stages of differentiation?
Yeah, we have, and in fact, thanks to Leah Boyle for helping us through some of our recent anxiety around this. In that, and I'll let Troy answer, but just briefly to answer your question, yes, we're seeing differences, and we don't know what to attribute them to yet, but the mutants line has been, well, we can't grow it yet, so honest. And Leah mentioned that their lab also had problems with this as well, and it took a number of rounds of trying to differentiate these cells from the iPSC stage to the neural precursor stage. So it does appear that something very early on in the development of these cells is awry. And so that just the other day, Troy and I learned this information, and I think that actually expands our experimental opportunities to look earlier in the development of these cells, to look at things like neurite outgrowth, and questions eventually in organoids that may help us understand the greater developmental profile of KAND affected nervous disease.
Great, and we'll do one with the time we have from Leah Boyle asking, are there specific cargoes that you're most interested in tracking once you get your system up and running?
Yeah, sorry, Troy, do you want to answer?
Yeah, I mean, certainly we're excited to start with BDNF and neuropeptides in particular as a starting point, looking at dense core vesicles as an actual, like the cargo that's being trafficked is certainly a starting point that we are very keen on examining thoroughly. Yeah, that's what we've planned out so far.
I mean, I can give you kind of a more mundane answer, just from the standpoint of the history of our lab. I mean, for a number of reasons, we've been interested in dense core vesicle transport for a long time. So really, it was just one of these serendipitous scientific events that we had been interested in axonal transport, sort of writ large. For practical reasons, we had an interest in dense core vesicles. To be totally frank, they're relatively easy to image compared to other cellular cargoes. And then through the years, we and others demonstrated that KIF1A is responsible for the transport of these neuropeptide-containing organelles. And we evolved, shall we say, into the KAND project. So it was, yeah, just luck in some ways.
Serendipity can be a really beautiful thing, especially in a scientific project.
Yeah.
But we are so happy to have you on board, and we look forward to seeing how this project develops over time. Thank you very much.
Thank you, and yep, stay tuned. We'll be back with more.
Sounds great.
Thank you.
Thanks.
All right.